Hello everyone, welcome back to this session once again. In today's part of the session, let me discuss some extent of the chemical parameters which we denote in terms of an organic matter consumed either through the biological degradation or through the aid of oxidation and reduction reaction. What are those kind of an oxygen requiring part of the parameters? They are namely the total organic compound and biochemical oxygen demand. In today's part of the session, I'll cover up only the total organic carbon and remaining BOD and all I'll cover up when I come across the usage of kinetic equations, because whatever the reactions taking place in a chemically oriented manner, they are to be measured in terms of an transformation of a substances where the reactants getting transformed in terms of an products basis of the external factors or maybe even due to the internal factors also. With this, let me take up even in the today's part of the session the biological parameters. I'll continue back the some set of the chemical parameters when I completely cover up the reaction kinetics. Let us take the chemical constituent, especially TOC, and in the previous class I have also explained related to the chemical oxygen demand. Chemical oxygen demand is a combination of the biodegradable organic matter and non-biodegradable organic matter. That is the one thing. Total organic compound, it mainly represents how much amount of the carbon they are present in the given sample. The determination of the total organic carbon, we call it as total organic carbon. The total organic compound, it is also another combination of the biodegradable organic matter and especially as well as even the non-biodegradable organic matter, whereas the result can be obtained within a span of 5 minutes through the aid of the instrument provided with a proper heating which initiates oxidation and reduction reaction along with the ultraviolet absorption medium. Ultraviolet absorption medium. They can be determined even through the spectrophotometric method, we call it as Fourier transform infrared spectroscopy. That is the one type of an instrument. In the combinations where the TOC analyzer is a special set of an instrument where you can get the value related to either hydrophobic organic carbon fraction or hydrophilic organic carbon fraction. These are the two different things. Whatever the different compounds that you are utilizing on a daily basis, everything contains one or the other organic compounds. For example, your toothbrush, shampoo, the soap, detergents, whatever the creams or maybe any lotions and all, it mainly composed of different combination of organic compounds. They may be either a trace organic compounds are also present or even the refractory organics also. Any cleansing agent you take in your home, you will get to know. Okay, for all those categorization we come across total organic carbon. There is also a special relationship between what is the BOD, what is meant by COD and TOC, what are those relationships. I will be explaining in the next part of the class. Now, in this session, let me take upon the biological parameters. Normally, In the wastewater, it is a home for different set of microorganisms. These microorganisms tends to play a very important role. There are broad type of categorization. Either if you take in the basis of the bacteria or viruses or fungi, these microorganisms are classified. Bacteria, virus, fungi, protozoa, rotifers, and worms. Let us take this particular parameter, bacteria. Where does the bacteria arise? Bacteria are unicellular organisms. They are ubiquitously present all over the world. No place where the bacteria does not tend to exist, means only in the some set of the cold regions. They are frozen in nature. And only in a special set of the conditions in the high temperature, we call it as thermophilic type of the bacteria. Mesophilic range means less than the thermal range, that is 20 to 45 degrees, and they are designated as mesophilic range of the bacteria. The bacteria are also categorized into three different types, basis of the oxygen consumption also. They are either aerobic in nature, where they consume or mainly depend upon the oxygen content present in the wastewater, and anaerobic in nature, anaerobic. In this particular type of an bacteria, they do not depend upon the oxygen content. They do not depend upon the oxygen content, they go for biological degradation. And there is no utilization of the oxygen. In the absence of oxygen, the bacteria tends to grow. That is the specialty case. One which is having the characterization of aerobic as well as anaerobic, we call it as a facultative bacteria. Example is our own mouth. During the daytime, saliva is proactive in nature, thereby the pH is maintained. Whereas, if you take in the case of the night times, what happens? The presence of the facultative bacteria tends to grow at a rapid rate. The rapid rate or the growth of the bacteria tends to create an odor. Okay? This particular set of the bacteria, we call it as a facultative bacteria. The aerobic bacteria mainly depend upon the oxygen having the carbon sources, and later on it is getting transformed. Later on it is getting transformed into the nitrogenous sources. Thereby, the transition part you get to know in the form of the facultative bacteria conditions. These set of the bacteria are very much important when we come across in the case of the secondary treatment, or we call it as biological treatment of wastewater, where the higher amount of the solids are degraded by the growth of the bacteria. And the bacteria which are beneficial, we call it as non-pathogenic in nature, non-pathogenic. They do not disturb, they are present in the intestines. Example, E. coli, they are the coliform group of the bacteria, E. coli. If the other set of the bacteria like anthrax or like bacillus, or maybe the different categorization, we have a spiral shaped type of the bacteria, rod, cone shaped type of the bacteria, maybe a regular rounded shape of the bacteria. cocci, streptococci, coli, coliform group of the bacteria. These are the subcategorizations coming across to it. And mainly, I would need to mention that the biological wastewater treatment has a vast area to learn. Only a limited portion you are getting to learn. If at all, if you need to come across for studying the microscopic biota studies and the toxicological risk assessment, the bacteria plays a very important role, and even for the precipitation as well as conversion of the ammonia to nitrogen gas, these bacteria plays a very important role, even for the phosphate also. If the bacteria are present in the living organisms, they tend to grow up and affect the host body. That we call it as a pathogenic bacteria. Pathogenic bacteria. Whatever the tuberculosis and typhoid, even in the case of the cholera, Vibrio cholerae, isn't it? These are the separate set of the organisms. During the wastewater, what happens? There may be the exchange of the gases. Along with the gases, the bacteria are driven by the wind action and expose it to the human body, or maybe any animals. They tend to attack and consume whatever the nutrients that are present. Okay. And this is related to the aerobic as well as anaerobic. There are different categorization, what are aerobic and anaerobic. And bacteria is also used as an indicator organisms. Indicator organisms to determine the extent of the pollution. Determination of the bacteria determination of the bacteria can be conducted in the laboratory. They can be noticed and observed by three different methods. One we call it as a pour plate method, spread plate method, that is one thing. It is normally done for the case of the fungi. Whereas multiple tube fermentation technique as well as membrane filter technique are incorporated for bacteria. Getting the point clear for this? I think you have studied the MFT as well as MTF also. This is a fine tuning for you to know the concept, for you people to know the concept. Virus. Viruses, they are the border between the living as well as non-living organisms. When it is inside the host, it tends to replicate and multiply it. And if it is at the outside, it is like a normal non-living thing. So this special set of the category has played a very important role. Recently, we have exposed to the larger impact on the economy due to the COVID-19 virus. Viruses, there are different types. Maybe the viruses which are affecting the bacteria, we call it as a bacteriophage, hepatitis type of the virus, polio virus, okay, polio, hepatitis, that causes even the jaundice also in infants, and the virus which tends to have an immediate impact even the water, it is even the coronavirus also. That is present even in the wastewater. Please do keep it in the mind. Determination of the virus, it is not a simpler task. When you need to sample, it is determined only through the basis of the reverse transcriptase enzymatic type of the reaction taking place in the laboratory cultural media. Depending upon that, you can come across. Whereas in the case of the bacteria, if you keep it in the colony counter and count how many number of the colonies are formed and express it in terms of an colony forming units per 100 ml. That is CFU per 100 ml. That is the normal part of the procedure. For the viruses, procedure is not enough. There are special instruments are to be required, and biological hazard is a major impact on the studies in the laboratory, especially the virology laboratory and microbiological laboratory. Fungi. Fungi, they are some of them are unicellular, some of them are multicellular. They are mainly the plant-like type of an kingdom where they do not utilize the sunlight. In the absence of the sunlight, they utilize only the chemical ingredients which are present either from the dead or the decayed organic matter. That growth, we call it as a fungi, either in terms of a mold or what we come across in the case of the mushrooms and all, normal layman terms and all. But this mold or the formations of the siliceous content or the fungi plays a very important role in the degradation of some of the mainly the biological characteristics if you take upon. And if I go for taking what is meant by the protozoa. Welcome back to the today's part of the session. In the previous class, I think we have discussed the concepts related to the biological parameters, especially the bacteria as well as the fungi. We had discussed all those set of the parameters. In today's part of the session, let us get to know much more concepts related to the type of the virus or the different instances where we come across the effects of the viruses. In the previous class itself only, I have mentioned that viruses are not only limited with respect to the spreading of the water, especially the diseases, even through the airborne. We call it as a waterborne disease and airborne. Now, in this particular case, we are dealing with the waterborne diseases. As earlier I have stated that it cannot be determined directly, it has to be determined through the reverse transcriptase enzyme, and based on the enzymatic type of the reaction, the viruses can or the strains can be identified which kind of the viruses or the protein exchange that takes place. This is the most fundamental thing that we need to keep it in the mind, and normally we cannot go for conducting or determining the virus concentration in the laboratory. It is very much hard to difficult or maybe difficult to analyze it. It is not there in for your syllabus, only extra information I am telling it. Now, the one remaining part which I need to discuss in today's class is related to the Helminth worm diseases. Helminth, or we call it as a guinea worm disease, like planaria and all those particular type of the multicellular organisms which are invertebrate in nature. They always depend upon the host or the living body for gaining much more nutrient and their growth. The formation of the egg and all in the host body, if it hatches, then the amount of the nutrient utilized for their growth is mainly depleting the host body. When it is depleting the host body, thereby the body immunity decreases, there will be abrupt changes in the physiological activities and maybe a vomiting, nausea and aversion towards taking of the food and all, dehydration. These are all the certain symptoms that tend to occur. Okay. This is mainly related to the Helminth type of the worm diseases. Next category we come across is protozoa. Protozoa, they are the unicellular organisms. Mainly there are different types of the protozoas available. Maybe you might have heard in your lower part of the classes, maybe in the high school or maybe in the few colleges in the biology, amoeba or entamoeba, and then cryptosporidium, okay, the cyst formation, all these things mainly, or trypanosoma, all these things mainly come across in the category of the protozoa. They affect even the bacteria, and they also affect even each and every multicellular organisms. They live inside the host of the organisms, but they do not tend to have any other external impacts upon the host body. They are called as a vectors or the carriers. For example, if you take mosquito, the mosquito mainly contains a plasmodium. The plasmodium type of the protozoa is mainly responsible for the causing of the malaria, or maybe even the dengue, like this, the fever that tends to come, because they are the carriers, those multiplication takes place. Getting the point clear? So, in this manner, whatever the protozoa that are present in the water, if it is not tend to treated in a proper manner, then it leads to the different sorts of the diseases and affects the human health and surrounding environmental conditions. Getting the point clear for this? And here in the protozoa, we can come across in such kind of an examples. Kindly observe some of the pictures that are being shown here. Okay. In the picture you can see what are the different types of the protozoas that are available. It may be entamoeba, histolytica, and the other set of the pathogenic organisms like cryptosporidium parvum and Blantidium coli and Cyclospora, entamoeba histolytica, Giardia lamblia. These are the major protozoas. They are pathogenic in nature. They are pathogenic in nature. What are the major symptoms? Diarrhea, and entamoeba histolytica, it is prolonged type of the diarrhea causing the damage to the intestine as well as even for the liver also. They damage the cell walls and all, thereby the tissues tends to get damaged, leading to the destruction of the endocrine system. Okay. And next important thing, it is mainly related to the cyst formations and all. They may be spherical, areas are responsible for the formation of the cyst. You might have heard about like the cyst formations, either maybe in the relative to the abdominal region, or maybe related to any of the region of the bodies and all. And cystic syndrome, for example, if you come across the polycystic ovary syndrome and all, the type of the diseases, not like a diseases, they are the kind of the category which they tend to accumulate inside the host of the body, but it is difficult to identify, and it looks like a normal itself only. It does not have any other things. Only when diagnosed in a proper manner, we can analyze it, or a doctor can analyze this. So, these critical parameters are the bridge between the engineering as well as the doctors also. Getting the point clear? Because public health engineering mainly deals with respect to the protection of the people. Who are the persons who need to protect the people? One thing, it is the engineers who need to take care of all these things, a microbiologist along with the doctors. So, they are the members who need to take care of spreading of the diseases to be curtailed and to give the remedial measures in order to see that the epidemic or the pandemic type of the diseases are to be declined. Okay. Now, this is related to the pathogenic organisms, and I think in bacteria also I have explained in the previous part of the class. So, next part of the category I have left out, that is mainly radioactive materials. Do you think that the radioactive materials are present in the wastewater? Obviously, only certain traces quantities are present, that is all. Majorly, it is measured in terms of becquerels or in terms of curies. These radioactive materials emit the two types of the radiations, alpha as well as the beta radiations. Gamma radiation, it is a neutral, alpha and beta radiations. What are those? Let us get to know. Mainly the radioactive elements, we call it as a radioactive parameters of the materials. You might have known that the phenomenon of spontaneous disintegration due to the unstable atomic nuclei with the emissions of the radiations, that phenomena we call it as a radioactivity. Whatever the elements starting from 85 to 92 or all or more than that, we call it as a radioactive type of an element. Okay, they emit the different radiations. What are those? Mainly alpha emitters, they are expressed in terms of a microcurie, mg per liter. Maximum, it is related to 10 raised to minus 7 in the case of the inland surface water, and in the case of the public sewers and to the land, we have 10 power 7 and 10 power minus 7. Okay. If it is in the case of the beta emitters, the negatively charged type of radiations, again in terms of a microcurie, so it is 10 power minus 6. These are the parameters, and if they are more than 7 or maybe 10 raised to minus 7 or less than that, if you take it as 10 raised to minus 3 or maybe 10 raised to 2, 10 raised to 1 and all, it is very much dangerous. Okay. Why it is? The reason, these radiation tend to cause an acute as well as the chronic effects. They also cause a cancer. Okay. These are the set of the parameters, maybe strontium isotope, uranium, thorium, plutonium, or lead extracted part of those things and all, they tend to cause the certain radiations which are harmful in nature. That analysis is only kept by the Central Pollution Control Board in order to maintain only to the minimum extent. And this set of the parameters are also highly tedious job, and it is very much difficult to analyze in the normal usages like atomic absorption spectrophotometer or maybe even the spectrophotometric analysis. They have a separate kind of an analysis, even the volumetric as well as the instrumental analysis. If any of the radioactive material is contaminated, then the whole lab is contaminated also, because upon the surface of emissions of the radiations, you cannot utilize all those glasswares or equipments and all. A separate room has to be kept for that. So these are all the critical things or the parameters to be kept in the mind. Next test, we call it as bioassay test. This bioassay test is mainly conducted upon for the fishes or maybe any similar kind of organisms. What happens? They tend to keep those organisms in an aquarium or maybe any of the required size of the material fabricated. The wastewater is introduced with certain dilution, and they go for checking whether the survival rate of the organisms are exactly 100% or any 90%, 40 or 50%. Based upon that kind of the test, we call it as bioassay test, means using the living organisms, especially mainly related to in the inland surface water, it is 90% of the survival of the organisms. In the case of 96 hours in effluent. And this holds good for both the inland as well as even the public sewers also. That is a major thing. Okay. Hence, these are certain set of the parameters to be considered. Moreover, we have larger, or we call it as a greater parameters. They are not easy to analyze, maybe a pesticide compounds and all. They require a special set of then equipments that we need to take it. Hope so you have understood all the concepts related to the characterization of the wastewater from the starting previous classes also. Now let me move on for the next kind of the topic, that is reaction kinetics. Why this reaction kinetics is very much important? What is meant by a reaction? What are those type of the reactions involved? Reaction means not only like a chemical equations. Reaction